Hello students, I hope that you all are fine. Due to this pandemic caused by coronavirus, our classes has been suspended for a while. But it is our responsibility to teach and complete the remaining course, and hence we have opted for online pedagogy. Being student of life science, I am expecting you to read about the coronavirus and its symptoms. You all should be aware of preliminary symptoms so that you can take preventive measures to keep yourself and your loved ones safe. In our earlier class, we had already discussed the environmental factors that affect the growth of microbes. We had conducted the group discussion, internet surfing and presentation to understand the topic. In today's presentation, I will introduce you to biological macromolecule or simply biomolecule. Before going to the presentation, let's see the content. The content for the presentation are introduction to biological macromolecule, difference between macro and micromolecules, introduction to carbohydrates, classification and its function. The first question that comes into our mind is, what are biological macromolecules? Biological macromolecule term has three words. First is biological, second macro and third is molecules. Biological means something which is related to life. Macro, which means large in size and hence it has high molecular weight. And a molecule, I am sure you must have read and learned this term in your chemistry class. If you try to recollect from your chemistry class, a molecule is defined as a group of atoms bonded together, representing the smallest fundamental unit of chemical compound that can take part in any chemical reaction. After understanding these three words individually, let's try to define it. So now, biological macromolecules are defined as a large complex molecule produced by living organism. There are four major types of biological macromolecule. First is nucleic acid, second carbohydrate, third lipid and fourth proteins. Let's see the difference between macro and micromolecule. As I said earlier, macromolecules, they are large in size. Usually these macromolecules, they are polymers of micromolecule. When micromolecules are linked to each other, they form a macromolecule. So the macromolecule like polysaccharide, nucleic acid, protein and lipids, they are made from simple sugars, nucleotide, amino acid, fatty acids and glycerol. The here image Laurel and Hardy. Macromolecules, they are large in size just like Laurel and micromolecules are small in size like Hardy. In this distinguishing table you can see that micromolecules are small in size and macromolecules are large in size. Micromolecules have low molecular weight whereas macro have high molecular weight. Micro, they are simple in structure and macro are complex in structure. Macromolecules, they are highly soluble in intracellular fluid whereas macro, they occur in colloidal state. The first biomolecule that we will study is carbohydrate. In general, the definition of carbohydrates is polyhydroxyaldehyde or ketone. The definition itself tells that it has many hydroxyl groups with either aldehyde or ketone as a functional group. In general, its chemical formula is CnH2ON. The primary function of carbohydrate is to produce energy. It also plays an important role in maintaining the structure of cell and it acts as a dietary fiber. After reading this point, let's see what are different sources of carbohydrates. The different sources are bread, banana, roti, rice and fruits. So whenever you eat all these kinds of fruits or veggies or bread, you get carbohydrate. Let's see its classification. In this classification flow chart, you can see that carbohydrates are majorly classified into three categories. First is monosaccharide, second oligosaccharide and third is polysaccharide. This classification is based on the number of monosaccharide or monomers. Let's try to understand them one by one. So the first is monosaccharide. Mono means one and saccharide means sweet. The most common example is glucose. In monosaccharide, the number of carbon usually ranges from 3 to 7 and hence monosaccharides can further classified as triose, tetrose and pentose or hexoses. As I said earlier, the carbohydrates are defined as polyhydroxyaldehyde or ketone. As per the definition, the carbohydrate have either aldehyde or ketone as a functional group. Monosaccharide with aldehyde group are called as aldose, and monosaccharide with ketone group are called as ketose. The example of aldose is glyceraldehyde, and for ketose it is dihydroxyacetone. Depending upon the number of carbon in monosaccharides, they are further classified as triose, tetrose, pentose and hexose. Here you can see structure of triose, pentose and hexose. The triose means 3. In glyceraldehyde you can see 3 carbon, and therefore glyceraldehyde is an example of triose. In pentose you can see 5 carbon, 1, 2, 3, 4 and 5, and therefore ribose is a pentose sugar. Then glucose. Glucose has 1, 2, 3, 4, 5, 6. Glucose has 6 carbon and therefore it belongs to hexose sugar. In this slide you can see here there is a linear structure of glucose which is forming a cyclic structure. Now how does a linear structure form a cyclic structure? The aldehyde, that is functional group present at C1, interact with the hydroxyl group of C5 and it forms a cyclic structure. As you know that the formula for carbohydrate is CnH2ON. So what will be the formula for glucose? Glucose has 6 carbon, so N will be 6. So it will be C6. If we keep 6 here out of the bracket and when we open the bracket, H2 will be multiplied by 6 and it will become H12 and O will become 6. So the formula, that is chemical formula for glucose, will become C6H12O6. The most common example of monosaccharides are glucose, fructose and galactose. Glucose and galactose have aldehyde as a functional group, whereas fructose has ketone as a functional group. Now let's see disaccharides. Disaccharides and trisaccharides belong to oligosaccharide. Di means 2 and hence such carbohydrates have 2 monomers linked by glycosidic bond. And while formation of such glycosidic bond or a linkage, water molecule is released. Let's see a few examples of disaccharide. In this slide I have mentioned sucrose, lactose and maltose. I am sure these terms are known to you. Let's see what sucrose is made up of. Sucrose is made up of glucose and fructose. The glucose and fructose, they are linked by glycosidic bond. Now from where you can get this sucrose? Sucrose is present in sugarcane, sugar beets, sugar maple sap, dates and honey. You can get lactose from cow's milk, goat milk, yogurt, cheese and ice cream, and it is made up of from glucose and galactose. And here again glucose and galactose are linked by glycosidic bond. Maltose is made up of from glucose and glucose. It has 2 glucose monomers, and wheat, barley, honey, pizza are rich in maltose. Now let's see what are trisaccharides. Tri means 3 and saccharides means sweet. Hence these trisaccharides are made up of from 3 monomers. Again they are also linked by glycosidic bond. So the examples for trisaccharides are raffinose and maltotriose. Students, one homework for you. You have to search that raffinose and maltotriose are made up of which monomers. You also need to search few different examples of trisaccharides. Now let's see what are polysaccharides. Polysaccharides, they are complex sugars, they are large molecule and they are formed from monosaccharide or monomers linked by glycosidic bond. If the polymers are made from same repeating unit, in such case they are called as homopolymer, and if the polymers are made from different type of monomers, then they are called as heteropolymers. So let's see a few examples of polysaccharide. The first example is starch. The starch is homopolymer. It means it is made up of from single or same kind of monosaccharide or a monomer. The starch is the second most abundant biomolecule on the earth, and the largest source for starch is corn, wheat, potato, tapioca and rice. Let's see structure. As I said earlier, starch is a homopolymer. So here in this structure you can see that all the monomers in starch structure, you can see 2 different type of structures. One is amylose and second is amylopectin. Amylose is a straight chain, whereas amylopectin is a branched. In amylose, the bond is alpha 1,4-glycosidic bond. It means the glucose molecule are linked to each other by alpha 1,4-glycosidic bond. Whereas in amylopectin, you can see a straight chain and a branch chain. So the branched glycosidic bond is alpha 1,6-glycosidic bond. Let's see second example. Second example is cellulose. Cellulose is the most abundant biomolecule on the earth. Cellulose is present in cell wall of plant cells. It gives structural support to the cell. Cellulose is also homopolymer and the repeating unit again is a glucose. But then what is the difference between cellulose and starch? If you remember, starch, the glucose molecule are linked to each other by alpha 1,4-glycosidic bond or alpha 1,6. Now the question must have come into your mind, then what is the difference between starch and cellulose? Because they both are homopolymer, they both are made from glucose, but here the difference is about the bond and branching. In starch, there was a linear chain and a branch chain, and it was linked by alpha 1,4 and alpha 1,6 bond. But here in cellulose, the glucose molecule are linked by beta 1,4-glycosidic bond. Let's see the functions of carbohydrate. The first function is oxidation of carbohydrates provides energy. It means whenever you eat carbohydrate, you get energy. It stores energy in the form of glycogen. Glycogen is also one form of homopolymer of glucose. Carbohydrate is a part of connective tissue. It also acts as a component for some hormones and enzymes. So here we have finished the first topic, our fourth unit. Please let me know if you have any question regarding this topic. Thank you.